Thank you. Uh, let's. It's my great honor to be here to present my talk. So my talk, the title of my talk is ProsperousPlus, a one-stop and comprehensive platform for accurate protease-specific substrate cleavage prediction and machine learning model construction. So protease-specific substrate cleavage is a very important. Protease specifically cleavage the peptide bond or target proteins, just like this. And this cleavage is a very important type of irreversible protein post-translational modifications. And this process is involved in many physiological processes, such as cell cycle pathway regulation and protein degradation. So the dysregulation of proteases is always associated with many diseases, such as human cancer. And the knowledge of protease-specific substrate cleavage is fundamental for our understanding of functional mechanism of proteases. So it is very important to identify the protease-specific substrate cleavage sites. However, experimental methods are usually expensive and also not available for many labs. So it is very important to develop the computational methods. Currently, so we conducted we systematically reviewed and evaluated 17 state-of-the-art computational approaches for the cleavage size prediction in our previous research. And these methods can be classified into two types according to their methodology employed. Generally, they are into two groups. One group is scoring function-based method, and another is machine learning-based methods. So generally, the approaches based on the sequence scoring functions are able to deliver the prediction outcome very quickly, while the machine learning prediction performance. So the scoring function-based tools rely on a limited number of scoring functions, typically just one or two scoring functions, such as Blossom 62, matrix, and PSSMs, etc. And therefore, they cannot comprehensively encode the amino acid sequence information for a given substrate. While the machine learning-based tools do not share this drawback, they require considerable time and availability of high-end computing hardware to compute and tune or optimize their predictive models. However, they are required to calculate high-dimensional features, and sometimes it cannot suitable for high throughput prediction. In the last few years, we also developed four computational framework for protease-specific substrate cleavage size prediction, including the ProCleave, which is based on the conditional random field and protein 3D structure data, and DeepCleave, which is the first deep learning-based methods to predict the caspase and MMP cleavage science. And also we developed a Prosperous, which is a high throughput predictor. These tools attracted a lot of attentions. For example, the web server of DeepCleave and ProCleave have attracted more than 30,000 job submissions from over 17 countries worldwide and more than 150 citations. And these tools can help biologists to identify the substrate cleavage science and generate biological hypotheses for biological validation. For example, ProCleave have successfully helped scientists to predict the cleavage science in the TMEM10B protein and enhance the biological analysis. And this research was published in Cell in 2022. And we also received many emails from the users worldwide, and they asked us to and many of us ask us to include more proteases in our tool, because most of our tool only covered less than 50 proteases. So we developed, so this motivated us to develop the ProsperousPlus, because currently we have a lot, over 110 proteases cleavage data in the MERPS database. So we developed a ProsperousPlus. And another motivation for us to develop the ProsperousPlus is that users cannot efficiently train models that target specific protease types, because most of users are biologists. They do not have the programming skills. So our tools are also motivated to provide a platform to facilitate them to build their own machine learning models. So this is the framework of ProsperousPlus, and you can see that the panel A shows the framework of ProsperousPlus, and include the developing of ProsperousPlus, including several stages such as data collection, sequence scoring, model construction, and evaluation. So in this study, we use the data collected from MERPS database, and we removed redundant sequences using the city heat with 17% as the threshold. And then we combine both the sequence scoring function methods and machine learning methods to build a two-level predictive system to leverage both advantages of sequence scoring function methods and machine learning methods. In this study, we employed 17 commonly used scoring functions in our study, and use the scores as the features to train a machine learning model. And in the model construction part, we proposed auto machine learning pipeline to which leverages our previous study, the advantages of our previous studies, and try to find the optimal, the machine learning model for each proteases. After that, we employed our trained models on our web server. And panel B shows the function modules of our web server. You can find that there are 3 function modules, including the prediction module, train your own model module, and use your own model module. In the prediction module, we deployed 110 models for 110 protease specific cleavage science prediction, substrate cleavage science prediction. And in the train your own model, we allow the users to upload their own sequences, and then they can use our own AutoML pipeline to build their own machine learning model. And then in the third module, use your own model module. They can use their trained models to conduct a prediction. And we also developed a standalone software package because some users are not willing to upload their own in-house datasets to the web server. So we also developed a standalone software. And this is AutoML algorithm. The main idea for this is to identify the the optimal models, including the some base models and also some three types of ensemble learning models. And so after that, we apply the popular k-means clustering with k equals 2 to evaluate the scoring functions. Because in this study, the features are the scores generated by 17 scoring functions. So we first want to evaluate how these scoring functions can help to discriminate both positive samples and negative samples. And in these figures, the different colors shows the different clusters generated by the k-means, and the different signs shows the different the true labels. For example, the dots means positive samples. So you can find that the result demonstrate that the sequence scoring function can produce well-defined natural clusters. So they provide a very good foundations for us to build accurate prediction models. So that we also evaluate these the models on the tenfold cross validation, and we can find that most of evaluated proteases can generate very good prediction result. With most of them are with average accuracy AUC values of to 9.63 and accuracy to 0.9. Yeah. And we also conducted independent test to evaluate our models. And the left figure shows the performance comparison result with ProsperousPlus and other state-of-the-art approaches regarding the three major evaluation metrics, including the accuracy, MCC, and F1 for 12 proteases on the independent test dataset. And the comparison result demonstrates that ProsperousPlus achieves the optimal predictive performance in most of the proteases. And the right figure shows the ROC curves of the ProsperousPlus compared with other state-of-the-art methods. And Prosperous performed best regarding the AOC with 8 out of 12 proteases, and the average AUC across all considered proteases for ProsperousPlus was 0.966, surpassing the second best AUC of 0.95 achieved by the ProCleave, and the third best AUC of 0.923 achieved by DeepCleave. So overall, all these results demonstrated that Prosperous achieved competitive predictive performance that exceeds the predictive performance of current state-of-the-art protease on average. So then we want to interpret which sequence scoring functions can provide it's more important for the prediction of ProsperousPlus. and so we use the sharp methods to generate the most important features for the prediction based on the independent test dataset. And we found that several features provide substantial contributions to the prediction of cleavage science across multiple proteases, including the the KNN, NNS, WLS, and IC50 features. So these four sequence scoring functions are arguably the key features that contribute to the cleavage science prediction by the models produced by the ProsperousPlus. And because you can see that KNN scores are identified by sharp as the most important features in 6 of the 12 proteases. And the WLS means weight, web logo-based scoring. Scores are also found to be useful and also achieve the best for 3 proteases.
And
I see.
Can you hear us? Sorry to interrupt you, but we have 1 minute left. If you want to get to the summary, that would be great.
Okay. Okay. So I will be soon. So after that, so our web server is freely available, currently free available and there, and source code are publicly available in GitHub. So in summary, we proposed ProsperousPlus, which includes 3 key features, which is provides the most comprehensive coverage of protease-specific substrate and cleavage science prediction, and it also provides state-of-the-art predictive performance compared with the state-of-the-art methods. And also, we provide both the web server and standalone code versions. Caters the need of different types of users, because some users would like to use the web server and some users are more prefer to use the source code. Thank you for your attention.
Thank you